Good morning, everyone. On behalf of students, faculty, and management of Bapatla College of Pharmacy and Bapatla Educational Society, we cordially invite you all for this day 2 of 1 week online international faculty cum skill development program on innovations in drug delivery system, organized by Department of Pharmaceutics, Bapatla College of Pharmacy, in association with the Sri Padmavati Mahila University, Tirupati, and the IPA Education Division. Now let me to introduce the profile of today's speaker, Dr. Rajesh Bapatla. Dr. Rajesh is the chair in Department of Pharmaceutical Science at Marshall B. Ketchum University's College of Pharmacy in Fullerton, California, USA. He did his B Pharmacy from Bangalore University, Master degree in Pharmaceutical Sciences from University of Toledo, and doctoral degree in Pharmaceutical Sciences from the University of Arkansas for Medical Sciences. He has over 12 years of academic experience with a special focus on accreditation, strategic planning, and faculty development. Dr. Rajesh has received the Teacher of the Award from 2 different universities. He was also elected as the Secretary for the Pharmaceutics section of the American Association of Colleges of Pharmacy. His research involves both the scholarship of teaching as well as drug delivery of controlled release dosage forms and analytical method development. He has extensively published articles, written book chapters, and presented several poster and podia presentation. We welcome you, sir. Dr. Rajesh, now you can share your screen and start the presentation.
Thank you, Mrs. Punjahalli, for a nice introduction. Sorry, I am just okay. Can you see me? Can you see us? All see the screen?
Yes,
okay,
sir.
Thank you.
Yes.
So, sorry, I thought I mastered it, and again it just went back a little bit. Anyway, thank you very much, Mrs. Punjuhali, for a nice introduction. And also, I would like to thank the Department of Pharmaceutics, Bapatla College of Pharmacy, and also Institute of Pharmaceutical Technology, Sri Padmavati Mahila Viswavidyalayam, and also Indian Pharmaceutical Association Education Division for providing me this opportunity to speak to you all. It's a great way of great way to teach students and also faculty, and also have a faculty development forum at an international level. And also, especially, I would like to thank Dr. T.Z.K. Murthy, sir, for conducting all this. And also, I would like to welcome all the faculty and students who are watching this Zoom meeting or webinar through Zoom as well as to YouTube. So as Mrs. Punjuhali mentioned, that I am a department chair in pharmaceutical sciences at Marshall B. Ketchum University in Fullerton, California in US. So today I'll be talking about modified release drug delivery, recent trends and case scenarios. Though title is a little long, first I would like to try to focus on modified release drug delivery systems. So what are modified release drug delivery systems, different types, and also I'll focus on the mechanism of drug release from these drug delivery systems. At the end, I'll try to focus on recent trends and also case scenario I'll be presenting at the end. So what are modified drug delivery systems? These drug delivery systems, they have a special type of release not offered by conventional release dosage forms. So when we talk about conventional release dosage forms, you should get into picture that capsules, tablets, these are regular conventional release dosage forms, right? So in a capsule and a tablet, without any modifications, without any adding polymers, just when you are adding drug and adding other excipients and preparing them into tablets and capsules, the drug release mechanism is driven by the properties of a drug itself, the physicochemical properties of the drug. Whereas if you try to change anything, you add any excipient or add any polymer to modify the release of the drug, to change the release of the drug. So to make it immediate release or to make it extended release, or you want to make it to delayed release, anything that you do to change the release of the release profile of the drug by adding something else, those are called modified release dosage forms or modified release drug delivery systems. So there are so many terminologies. Sometimes it is so confusing. I'll try to give you the best shot that whatever I came across in a simple way. So let's start with the delayed release. Delayed release, anything that releases at a time other than promptly after administration. Say for example, you don't want the drug to be released for 4 hours, and you want the drug to be released only after 4 hours. Then that's called delayed release. We take the food, it is processed by digestive system, and then it will go into our stomach, and there it will stay for 4 hours. Then it leads to small intestine, right? So we don't want any drug to be released in the stomach, so we delay that for 4 hours. That's a delayed release. Now let's move to extended release. In an extended release, it allows at least twofold reduction in dosing frequency. So normally when you take amoxicillin capsules or tablets, normally the physician writes that the patient should take 3 to 4 times a day, meaning that every 6 hours or 8 hours, right? So you're taking 3 or 4 tablets a day of a tablet. So instead of taking 3 to 4 times a day, if you are taking only once the tablet, then you are reducing the dosing frequency to once, from 4 to 3 to 1. So that's called extended release. So instead of giving 4 times, you are giving only once. That's extended. Now let's move on to prolonged release. In a prolonged release, it's nothing but it releases the drug for absorption over a longer period of time. So instead of 24-hour drug release, we wanted to continue the drug release to be over 48 hours or 72 hours or 1 week. So same amount of the same drug, it can be same release rate, or it can be decreased or increased, but we want the drug release to be prolonged for a longer time. That's a prolonged release. Now let's move on to repeat action. In a repeat action, it contains 2 single doses of medication, one for immediate release and one for for a delayed release. Or it can also say that you have one type of drug A, you want to be released in the morning, drug B, you want to be released in the night. And then you follow the pattern on the next day, and the day drug A, drug B on the night. It's repeating its action. Daytime only drug A is releasing, nighttime only drug B is releasing, and this pattern is repeating. This is also, it is known as repeated action or pulsatile. Now, targeted release, I don't think I need to mention too much about it because it's targeting to a particular cell or a tissue or a component of a cell. If anything, we are specifically targeting to a particular location, that's a targeted release. Now, let's talk about controlled release. That's one of my favorite because I did my PhD in controlled release. So that's my favorite topic. So in a controlled release, it is denied to release the drug in a controlled manner. So everything we control. Either it can be a rate. So if we want to deliver the drug for, let's say, 25 milligrams per hour for a longer period, every time we want the drug to be released only 25 milligrams, not more, not less. That's a rate control. Duration. how long we want to control. So that's a duration, and then location where we want the drug to be released. It could be stomach, could be intestine, or any specific target. Sometimes these are overlapping control release with others, but I just control release has a specific definition, so I just put it there. Control release can be temporal, where we have a control over rate of a drug release, or it can be spatial, where we have a control over the space of the drug release. So having said that, we want to now there are so many modified release delivery systems or control release systems available, but if we discuss all of them, we will not have enough time for today. So I am limiting my talk to oral modified release drug delivery systems. So this picture, probably majority of you are familiar with it. Some of you have studied in your B Pharmacy. Later on, it's master's and PhD levels, all the levels you've seen either in pharmaceutics or pharmacology. But I'll just give you a brief explanation of what I'll be alluding to. So on the slide on y-axis, you have plasma drug concentration. So the concentration of the drug in plasma on y-axis, and x-axis you have time in hours. There are two horizontal lines. Each the second horizontal line that represents minimum effective concentration, and the top horizontal line represents minimum toxic concentration. So anything below MEC, the drug will not be effective in the body. It cannot have any effect. Anything above MTC, it will be too toxic to the body. So ideally, our drug release should be in this window known as therapeutic window. I'm sure all of you are familiar with that. Now, let's see what will happen if you have conventional release dosage forms such as tablets and capsules. So a patient takes a tablet, and as soon as the patient takes a tablet, sometimes depending upon the drug release profile, the patient may have to take the tablets 3 or 4 times. In this case, I would assume patient is taking 3 times. So each time the patient takes the tablet, it will reaches the peak, almost touches the toxic level, and then goes as the body distributes and eliminates, the drug concentration goes down. And if you just leave it, if the patient doesn't take the next tablet, so eventually the concentration will go down, right? So in order to keep the drug concentration in this range, the patient have to take the second tablet and then continue with the third tablet. So we will see peaks and troughs in the drug release. There will be fluctuations. That's normal with a conventional release dosage form. Now with a controlled release dosage form, so the red line that you see in the center, once it reaches a certain concentration, it continuously releases the drug, same amount of the drug over the assigned period of time. Say it could be 1 day, 1 week, or 1 month, depending upon your profile. So by seeing this, we can understand that we can reduce the fluctuations in the blood by using controlled drug delivery systems. Now let's move on to the advantages of controlled drug delivery systems. The controlled drug delivery systems reduces dosing frequency. As I mentioned in the initial slides, instead of taking 3 to 4 times a day, we can take 1 tablet a day. That will really help the patient, because the patient can easily remember. If the patient have to take only 1 tablet a day during night time, the patient easily remembers that. Reduction in blood level fluctuations. So we just discussed, instead of having to go through peaks and troughs, there will be smooth blood release profile, so there won't be any fluctuations. That leads to ultimately overall less side effects, because we are controlling the drug release. Drug will be released in the therapeutic window. There are no toxic effects or no minimum effective concentration, so it will have a reduced side effects. There is a reduction in overall healthcare cost because the patients, they'll comply with the medication more easily than compared to regular dosage forms. And also it reduces the frequent hospital visits. So there will be a reduction in overall healthcare. Though controlled release dosage forms are much more expensive than conventional release dosage forms, when you look at the overall picture, it is much cheaper. And obviously, it is more patient compliance. As I already alluded that the patients, especially consider the elder patients or geriatric patients, say our nanamma, tadaya garu, or nani, dada, dadi, they're old. They're, let me say, assume that they're 70 years or plus. And if you ask them, they have 2 or 3 comorbidities. Say they have a blood hypertension, uh, diabetes, and other conditions. For each of them, if they have to take 2 or 3 medications, and each medication, if they have to take 3 times a day, in a whole, they'll be taking 6 to 9 tablets a day, every day, and they have to repeat that for a longer period. Instead of that, if we can give a tablet that can last for 1 week or 1 day even, that will really help the patient. They will remember that, they will do better with their medication. Coming to the disadvantages, I think with some of the controlled release dosage forms, especially with the reservoir system, which I'll be talking in a later slide, there will be, because in a controlled release dosage forms, we will have more amount of the drug that is contained in a tablet that is supposed to release over a time. But if something happens, like accidentally patient chews the tablet or break the tablet by chewing in mouth, entire drug will be dumped immediately or released immediately, and that leads to toxic effects in the body. And also GA transit time is limited. I mentioned a lot about we can have 1 week time, 1 month time, especially if you have a oral release dosage forms, tablets, and you want to deliver it for more than a week. But our GI system is very limited, meaning that food can stay in the stomach only for 4 hours, right? And then on top of that, it will go to the small intestine, stays there for 4 to 5 hours, and then it goes to large intestine. There, it's depending upon individual's metabolism, it can stay from 10 hours to maybe 1 or 2 days. So though we can design a drug that can stay, that can continuously release for a week, but our body cannot keep the drug or dosage form in the body. So it will be washed with the food, and it will be coming out through feces. Another disadvantage is the reduced potential for dose change or withdrawal in the event of toxicity, allergy, or poisoning, overdose. So if something happens, or if the already tablet is in the body, it's really hard to take it out. And in a conventional release dosage form, after 8 hours, the effect of the tablet should wane. But with the controlled release dosage form, if we are designed the tablet to be there for a longer periods, then that we have more amount of the drug that is in the body. In case of allergy and in case of toxicity or overdose, that will be very lethal. So far I just gave an introduction about modified release dosage forms, and especially we talked about oral drug delivery systems and the advantages and disadvantages. Now we are reaching into key area. This is where I'll be spending most of my talk about different types of controlled release dose systems based on their mechanism of drug release. The first one is dissolution controlled system. So all of you are familiar with probably the word solubility. So if a drug has a low or poor solubility, what will happen to dissolution? Normally, it will have a lower dissolution, right? Dissolution is when a solid drug goes into a solution form. That's a dissolution. You have a solid drug. The solid drug has to get into solution in order for it to be absorbed. The process is called dissolution. If the drug has a low solubility, it doesn't want to get into solution, then dissolution time takes a longer. So normally the dissolution process, the solubility of the drug itself defines the drug release. So majority of the case, that is what happens in poorly soluble drugs, poorly aqueous soluble drugs. The next one is diffusion controlled. In a diffusion control, what is diffusion? Diffusion is movement of molecules from a region of high concentration to region of low concentration. It can be through a semi-permeable membrane in case of drug delivery systems. There are two types of diffusion systems. There are monolithic and reservoir. Monolithic are also known as matrix, and reservoir we will discuss about this in next slides. The third type is ion exchange resin system. So in this system, there will be a transfer or exchange of ions between the resin and drug complex with ions in our body. Our body contains calcium, potassium, magnesium, and these are positively charged, and chloride. These are different ions we have in our body, right, at different places. These ions, they will come and attack the ionic complex between the drug has to be charged drug, and resin. Resin is oppositely charged functional group. It is attached to it. So ion exchange between the charged drug and the ions in the body, that will take place. The fourth one is osmotically controlled. As the name suggests, osmotic pressure created by osmotic agents in the delivery system, that will exert pressure. Because of the pressure, the drug is in solution form will come out through a hole that is made. Erosion control, it's a polymer that erodes upon ingestion. And finally, environmentally controlled systems. In environmentally controlled systems, the environment itself controls the drug release, not the delivery system. Whatever we talked so far, mostly drug release system, the delivery system that controls, but here the environment that is surrounding the delivery system, that is controlling it. So we will go in detail about each of them. So far, is the audio clear, or do you need me to?
It is clear, sir.
Okay, thank you. So the first one is dissolution controlled. In a dissolution control, as I mentioned, it employs the drug dissolution as the rate controlling step in the drug release. So normally drugs, they have, like if you look at the physicochemical properties of the drugs, they have a solubility as one of the parameters, and they have the solubility is innate property of the drug. You cannot change the solubility. You can change dissolution, but you cannot change solubility. So if the drug by itself has a poor solubility, that can act as a dissolution control. I have a few examples here. There are many examples that you can look in the literature. These are the drugs that can act as a naturally controlled release products. So the next one is diffusion control system. In a diffusion control system, the release rate of the drug is dependent on its diffusion through inert membrane barrier, usually water-soluble polymer. So as I mentioned previously, in a diffusion process, drug moves from a region of high concentration, which is the delivery system, in the such as tablet. Tablet has a high concentration of the drug. A drug moves from the tablet, passes through a polymer membrane. Either polymer is uniformly distributed or it is surrounded by a polymer membrane, and then comes out of the dosage delivery system into the body. In the body where the drug concentration is very low or minimal. So drug moves from a tablet into a body through polymer system. It will diffuse out. It will not dissolve. It will dissolve, but it has to, after dissolution, it has to diffuse through the polymer membrane to come out. It's a diffusion control, and there are two types. First one is matrix, and second one is reservoir system. In a matrix system, the drug is dispersed homogeneously throughout in an inert polymer matrix. So you have the drug and you have a polymer, you mix them together uniformly, and drug is dispersed everywhere. Drug is surrounded by polymer, but drug is not enclosed by a polymer. So in a drug in the outer layer, first is exposed to water. Whatever the drug that is on the outer surface of the tablet will be exposed, and water dissolves the drug, and drug will come out, leaving some pores in the tablet. So water enters through the pores into the tablet, and then again dissolves more drug, and the drug will come out. The dissolved drug will come out, and the process continues until the water reaches the center of the tablet. Here in a matrix system, initially you have a larger surface of the tablet. You have a large surface, right? You have a big tablet. You start with a big surface, larger surface area. As the time progresses, the surface area of the tablet becomes smaller and smaller. So with the matrix system, it's not possible to have a zero order release, because in a zero order release, you have to have a continuous amount of the same amount of the drug all that to be released all the time. Whereas with the matrix system, initially you have a large amount of drug because the surface area is big. More drug is coming out. As the time progresses, the surface area becomes smaller and smaller, so the drug release gradually reduces. Now let me show you a cartoon about drug diffusion controlled matrix system. There are two types here depending upon the type of the polymer that you use. So here it's a tablet, representative of a tablet, and the black dots on this tablet, where that's a drug, imagine that's a drug particles, and it is surrounded by polymer. The red space, that wherever you see, imagine that's a polymer. And now we have a water insoluble polymer or lipophilic polymer. So the polymer is made with a lipophilic material. It doesn't like water, so it doesn't dissolve in water. So whatever the drug is present outside, it will be dissolved and released. And then through the pores, water enters and release more and more amount of the drug. Eventually, you will see what you're seeing here is entire tablet. It remained intact, majority of it, because the polymer is not dissolved. The drug that is present in the polymer is already gone. So sometimes you will see a ghost tablet or an empty shell, what we call. So patients sometimes may come back to the physician and saying that, oh, doctor, I have taken the tablet, but everything came through feces. But as I'm teaching here to pharmacy students, so I would teach them, this is what this is what you should explain to the patient. No need to worry about the drug delivery. Everything is released. It's just a polymer. It's just a empty shell. Now, same matrix system. Consider using a water-soluble polymer or biodegradable polymer. As you can see here, along with the drug, some polymer is also degrading. You will see spaces here, white spaces in between here and there. That's a polymer also degrading. Over the time, all the drug will be degraded, and also the polymer also starts deteriorating. And you'll see this here, and then eventually entire tablet disappears because it's a biodegradable polymer. But this is a matrix system. Now let's talk about the advantages of matrix system. In a matrix, it is easier to produce than reservoir system, because in a matrix system, all you need to do is you mix the tablet, the drug, the polymer, and other excipients, mix them together, mill them, and do all the regulatory, whatever you do for regular conventional tablets. Just mix them together and make a tablet. You don't have to have a special equipment or anything like that with a control matrix system. And it can deliver high molecular weight compounds. So here the drug is surrounded by polymer, but it's not enclosed by a polymer. So polymer the drug, as soon as water comes, drug can dissolve, and through the spaces it can come out. There is no so with the reservoir system, the high molecular weight compounds cannot be delivered. Now the disadvantages, as I mentioned, we cannot obtain zero order release because I explained with surface area. As the time goes on, surface area decreases, so less and less amount of drug is released. But in my PhD project, this is a project that I have to do. With the matrix system, I have to provide a zero order release. Luckily, I was able to do it. I was able to defend it. I showed that when you are using different types of polymers, you can still have a zero order release when you use. I made a I made a drug excipient complex. Through the complex, slowly drug is releasing, and then I used a very water soluble polymer. So we were able to do that. Hence, I got my doctorate. So additional disadvantage is that removal of remaining matrix is necessary for implant systems. So if you have a lipophilic polymer, it doesn't dissolve in the body. Sometimes you may have to surgically remove it. That could be another disadvantage. Now let's talk about controlled diffusion using a reservoir system. It is characterized by core of the drug. And so you have a drug core in the center. It is surrounded by a polymer. So you have a drug core in the center. In the previous one, the drug is distributed uniformly, whereas here, drug is in the center, drug core, surrounded by a polymer membrane. The nature and thickness of the membrane determines the rate of release. So if you have a lipophilic polymer, the polymer is lipophilic, that is covering the drug, then water cannot pass through and drug release will be very slow. Whereas if you have a hydrophilic polymer, water can easily pass through, hence faster drug release. That's the nature of the polymer membrane. Thickness, the thicker the membrane, the slower the drug release, right? It's a common sense. Now let me show about the controlled reservoir system. So this is a red, the blue colored, everything, whatever you see inside the circle, this is all drug core. And the polymer is only under the black one, the black ring that is surrounding the blue color, that's the polymer membrane. So polymer is only present on the outer surface, not inside. Okay, like a regular process, the tablet is taken by the patient, it dissolves, and so water enters through the outer semi-permeable polymer, and then drug is dissolved, and as the time progresses, more and more drug dissolves until all the drug is released. Now, after some time, depending upon the type of polymer, if it is the polymer that can withstand, that can stay for maybe a day, and then after that it will disintegrate, then you will see the entire tablet disappears. But if you have a lipophilic, if you have a high molecular weight polymer, then you may see a tablet shape in the feces. So at this time, I will pause for 1 minute or a few seconds, and I want you to think about scenarios in a common scenario to explain difference between matrix and a reservoir system. Like now we have COVID situation, pandemic situation. You're all at home. You're having nice food. I know some home food. Some sweets. Think about some sweets that represents matrix and reservoir system. So here in US, the example that I give, and I will give you the Indian example. Especially, I'm from I speak Telugu in my house, so I'll tell some of the examples. But here in US, we use chocolate chip cookie. So it's like a cookie or a biscuit, and it has chocolates embedded, surrounded by cookie dough. And then probably you have seen gems or M&M. I think India it's called gems. So in both chocolate chip cookie and in gems, imagine that chocolate is the drug, right? So you have the chocolate, that's a drug. In chocolate chip cookie, the chocolate is spread out, dispersed in cookie dough or biscuit maker dough. So it is a matrix system. the drug is surrounded by polymer everywhere, and polymer drug, it's all uniformly mixed. In gems, you have the chocolate at the center and covered by a membrane. So that's a reservoir system. Now I was asking my wife, because she's in charge of the cooking and home, about some examples. So she told me, when I explained the scenario, she told me, so first let me talk to Telugu audience here. So she said kajikaya. Kajikaya. So it's made with coconut, and then it mixed with gourd or bellam, and the central portion you have coconut is a drug that's mixed with some ingredients like jaggery. Then it is covered by maida or something. It's like a dough. So that's a reservoir system. So, and copper laddu. I mean, it's just like coconut laddu. Just laddu mixed with some things. Those are like coconut is, that's a matrix system. In Hindi, so it's a coconut cookie or coconut biscuit. That's an example of matrix system. And gujia, that's what I'm not familiar with Hindi words, but I had to ask my colleagues here. So they said gujia is the word for that one. So gujia is the your reservoir system, and then coconut cookie or biscuits, that's a matrix system. I hope you understand. Once I explain this, my students actually, they really love it. They say, "Oh, Dr. V, I just remember now. I will never forget it." So that's the difference between reservoir system and matrix system. Coming to advantages of reservoir system, zero order delivery is possible because the only thing that controls the release is a polymer membrane, and polymer membrane always remains the same. You will not change. So same thickness, same nature. So zero order is possible. Release rate variable. If you want to have a faster release or slower release, all we need to do is change the polymer type and thickness. The disadvantages, the potential toxicity if system fails. If in a reservoir system, you have a drug core surrounded by a polymer, if small break in the polymer, just somebody accidentally patient bites, so entire drug will be released. There is no protective mechanism. Whereas in matrix system, even if you break the cookie, the polymer, the drug is cookies, chocolate is still covered by polymer everywhere. So still it is controlled. Whereas it is with the reservoir system, it's really difficult. High molecular weight compounds cannot pass through because the drug is inside, it's a high molecular weight, and the polymer membrane, it has to pass through the polymer membrane, but it is difficult because if it is too high, polymer membrane will just hold them. And cost is associated with uh, manufacturing is high with the reservoir system. Next is ion exchange systems. In ion exchange systems, the ionic drug bind to the oppositely charged functional groups of resins to form ionic complexes. So drug release is mainly by ion exchange. As I mentioned, for example, you have we have a resin. To the resin, we attach negative functional groups such as sulfonate functional group, SO3 minus. Sulfonate functional group is attached to the resin. Now you have a positively charged drug such as diltiazem hydrochloride, propranolol hydrochloride, any positively charged amine functional groups you have. Because of the positive and negative charges, positive charge of the drug, negative charge of the sulfate functional group, they immediately form ionic complex. So they have ionic attraction, positive and negative, they like each other, so they'll form ionic bond. And then from the resin and drug complex, you make a tablet, and the patient takes the tablet inside. Once the tablet is in the body, our body has, as I mentioned, calcium, potassium, sodium as a positive ions, chloride as a negative ions. So these ions will come and attack the ionic complex between the drug and the ion resin, sulfonated resin. So the magnesium or calcium will come and attack, bind to the sulfonate function group, releasing the drug. So drug will be released in the body. Ions exchange. This is an example. This is a type of delivery system, controlled delivery system. Excuse me. The advantages of high drug loading is possible because in a resin you can have so many functional groups. Sulfonated functional groups. You can add so many functional groups. To that, each functional group you can add so many drugs. So at least one functional group, one drug. So you can have a high drug loading. pH is independent only with a sulfonated functional resin group. Whereas if you are using carbonated carboxylic functional group, then it depends on the pH. The disadvantages: severe tailing at later stage of the release. It has so many drug molecules attached to it, so keeps on releasing the drug for a longer period of time, and you see slow decrease in the drug release. That's a disadvantage. Now let's move to osmotically controlled release system. In an osmotic control release system, this is an example of Oro system developed by Alza Corporation. Now it's part of Johnson & Johnson. So it is like a reservoir system. Only difference is it has a hole, right? See, you have a drug and excipients inside and surrounded by a semi-permeable membrane, which is nothing but a reservoir system. The only difference here is an osmotic, precisely drilled. So the size of the hole is very precisely drilled by using laser. We use a laser to drill the hole here. So based on the size of the hole, the amount of the drug will be released. So here, the black ones, the green one is the drug, black one is osmotic agent. This is also another difference between reservoir system and here. In osmotic drug delivery system, we have osmotic agents such as sodium chloride, mannitol, or other menthols. There are some osmotic agents that can be used. They are incorporated. And you have a semi-permeable membrane, as I mentioned, the pink one. And then as the patient takes the drug, the dosage form, water gets inside the osmotic tablet or pump through semi-permeable membrane, and the drug absorbs water and it will dissolve. And at the same time, osmotic agent that you have, sodium chloride or mannitol, they also absorb water and they starts expanding. They will start creating osmotic pressure inside. So there is a pressure building up inside the osmotic pump. Because of the pressure, whatever the drug that is dissolved, there is only way for the drug to come out is through this hole. So the drug is coming through this hole in a liquid form through the hole that is precisely drilled. So at any given point of time, only that amount of same amount of time, same amount of the drug will be released through the hole. And it doesn't depend on the environment. Environment has no effect on this. Wherever it is, all it needs is water to get in, and osmotic agents will absorb water, create pressure, and push the drug out. There is a modification in this one is they came up with Oro's push-pull system. The name push-pull is, there is in addition, now in the previous one, the drug and osmotic agent, they are mixed together in one compartment. Whereas here we have the drug compartment is separated by separated from osmotic agent compartment through a semi-permeable or movable portion. It's a polymer that can move. So as soon as the water enters, osmotic agent creates osmotic pressure. Because of the pressure, this movable portion will move. It will move forward, push. It moves up. So the drug in the solution form, it will be pushed up. So the drug in this compartment will come out. That's the only difference between the previous one and this one. So osmotically controlled systems, these are special type of reservoir systems into which osmotically active agents are incorporated. I mentioned sodium chloride, mannitol, these are examples. So osmotic pressure as the driving force to release the drug at a constant rate. That's in a reservoir system, it's just the polymer membrane that is controlling. Here, osmotic pressure, that is the driving force. So you need to, though they look same, they are similar. The difference is this: osmotic pressure is the one that is a driving force in osmotic control systems. So advantages: release of a drug is independent of environment of the system, because wherever you put, it doesn't matter. As soon as water gets in, then osmotic pressure is created inside, so drug will be pushed out. G-rod release is possible because it's just through the hole it has to come, and hole will not expand or contract. So all the time same amount of drug will be released. Disadvantages: they can be very expensive because it's it's not easy to precisely drill a hole and a small tiny tablet or a capsule, and quality control is also more expensive. The next one is erosion control system. So in this case, it's a polymer that erodes over time. So it's similar to what we discussed already in matrix system. It's a hydrophilic polymer. So you have the drug surrounded by a polymer membrane, polymer uniformly distributed, and as the time goes, the drug and the polymer starts eroding, and you'll see polymer also degrades, and eventually all the polymer and the drug that completely erodes. There are two types. It's a bulk erosion and surface erosion. In a bulk erosion, the water rapidly penetrates and entire dosage form and polymer degrades throughout the matrix. Whereas in surface erosion, only at the surface the polymer starts erode, and then eventually it will go into the center, and everything will be eroded. So the next one we will talk about is environmentally controlled systems. Here, the environment in which these delivery systems they are placed in, depending upon the environment, these delivery systems change their shape. They will release the drug. So it depends upon the environment, they will act. So the polymers, these type of polymers such as cholesterol hemisuccinate, these are examples of polymers that can sense changes in pH or temperature. They can swell or shrink or they erode depending upon the change in environment surrounding the delivery system. Say a patient who is suffering from cancer, right? So in a cancerous conditions, what will happen is the cells surrounding the cancer tumor, there will be acidosis develops. So there will be the pH of the environment next to the cancer cells actually goes down. We can design a polymer that can sense the change in pH. So under normal conditions, this polymer remains as it is intact. As soon as it reaches the cancer site, the pH of that area is actually goes down. So this polymer identifies, okay, this is pH is changed. That's where it starts opening up or changes conformation. It relaxes, or whatever it does, the drug is released. Or in other case, there is an inflammation. Normally, if you have a fever or in any conditions, there is an inflammation. In that inflammation, there will be increase in temperature. So increase in temperature, certain polymers, they can change, they can sense the change in temperature, and then accordingly they will release the drug. So I have a cartoon here that shows it is a normal tablet with a polymer and the drug, as you can see. As it reaches the different environment, what will happen is the polymer is expanding now, right? It's relaxing. So there are more pores open up, and the drug has more space to release. So that's how drug releases, depending upon the environment. Or we can also do the same thing. We don't want the drug to be released in certain cases. So we can design a polymer that shrinks actually at that place like this. So it is constricting the drug even more. It's not releasing the drug. So these are depending upon the environmentally controlled systems. And this will be probably my last type of system. So this is a polymer. It's that has glucose oxygenase enzyme. So probably some of you know what is the purpose of glucose oxygenase enzyme. It converts glucose into glucuronic acid. And this polymer, inside the polymer, it contains insulin. So insulin is trapped inside the polymer that is actually in a relaxed condition. But the polymer here, they have glucose oxygenase enzyme. Under normal conditions, the pH of the blood will be around 7.4. Nothing is going to happen. It will be the insulin is trapped inside. It will work. The dosage form will be traveling inside the body. As soon as more glucose is released, this glucose is extra glucose is sensed by the glucose oxygenase enzyme. So this enzyme converts glucose into glucuronic acid. So what will happen to the pH once we convert glucose into glucuronic acid? pH goes down. So at lower pH, these polymers, they constrict. They sense a change in pH, so they constrict. So the insulin is released. So it's like on demand, insulin can be released. This is an example of environmentally controlled systems. So that I have normally in my class here in US, I will be giving a case to the students based on the presentation, and they will be working with their groups, and they will solve this with the groups, and they have to post me the answers. Since we don't have groups here, we have a large one group. I would just highlight the case, and then I will go through the answers. So the patient is has significant neurological deficits, and then he's recovered. Right now, he's receiving extended release tramadol for chronic pain. And last week he had a stroke. So because of the stroke, he had a gag reflex. So anything that he eats, that he'll throw up. So basically, he has to feed through a gastric tube. So on July 17th, a patient's medication switched to gastric tube route because he cannot swallow. All dosage forms have to be crushed. Remember, this is an extended release dosage form, and it is a reservoir type. All dosage forms have to be crushed and administered through gastric tubes. So the nurse, she did not know that it's a reservoir system or something. So we crushed the medication and fed him through the gastric tube. So that leads to some somnolence and disinterest later. And also the pain was not reducing as it is intended. So these are usually my questions for the students. So anything wrong with the order, and what is a possible explanation for the note? So these are the things I will be asking them, and they will be answering it. So I think that comes to end of my presentation. I may be slightly over the time. So yeah, yes, sir.
Yes, sir. Thank you very much, sir. Now, may I request Nagaraju sir to moderate the questions.
Good morning to all of you, especially my good evening to Dr. Rajesh, Bapatla. A very nice presentation, a very lucid manner, where these both graduate and postgraduate students can easily understand basic concepts. And also the final slide, I really like the case studies, which really at the end of the session, if the opportunity is given any students to write, we can really understand how much the topic has gone to their brains. And it is very nice throughout the presentation. I was listening to you. and it is a great time to spend in the morning in India with you. We have
Thank you, Dr.
almost
Nagaraj.
3,000 plus participants watching in the YouTube, and also around a few hundred watching in the um, Zoom app. And we have
Good.
plenty questions, number of questions dashing us to clarify with you. But however, due to the time limitation, I'll be focusing only few questions, and where our team, Dr. Satish Kumar and other people, have edited the few questions and merged few questions to similar questions to it. So any questions, if people are watching, the students, especially the young researchers, you have any clarifications regarding this, we can join us at the panel discussion at the end of the FDP program. If possible, Dr. Rajesh may also join us to answer your questions, or otherwise, those questions can be, we will pass it him over the mail or WhatsApp, so that we will get back the answers for the benefit of the young researchers. And the coming to the questions, people are asking, how do you calculate the dose for the controlled drug delivery systems?
So that's a great question. So first, normally like any regular dosage forms, when the manufacturing unit, the manufacturing companies, they do first studies on clinical trials. So they will determine in I think phase 2A, phase 2B, what is the actual amount of the drug that is needed. So normally the controlled release dosage forms, they come after the conventional release dosage forms have been already released. They won't release directly. So already the manufacturers have an idea like how much conventional dosage form that the patient is receiving. Based on that, so if a person is receiving 3 times a day, three tablets a day, and if the tablet is 500 milligrams per day, then it's 1500 milligrams per day, and we design in such a way. I think, but still, whatever we do in pre-formulations and formulation stage, again, it has to go through clinical studies. They have to do the bioequivalence studies to justify that. But at this point, usually I spent my career in only in academia. I'm not in, I mean, I worked in industry like back in India in Aurobindo Pharma, but I was in regulatory affairs and never been in formulations at that time. I have a limited knowledge, but maybe people like you have a better answer than that. I think that's what I have at this point. Sorry.
Another question, what is actually the scope of pH independent controlled drug delivery systems?
Could you repeat that again, please?
pH, can you just put some light on pH independent drug delivery systems, controlled drug delivery systems?
Okay. So we talked about pH dependent drug delivery systems, and especially the polymers, they can sense the changes in pH and they will release the drug. There are some delivery systems that they won't change based on the pH. pH doesn't matter for those. Those are called pH independent delivery systems. In my slides, I mentioned about osmotically drug delivery system. That's a pH independent drug delivery system. And also even in matrix system, depending upon your drug and the excipient that you're using, if the drug is very strongly bound to your excipient, it doesn't matter where the pH is, and mainly it is controlled by the polymer and also the change. Reservoir example of a pH independent is, I would say, osmotically drug delivery systems, that it doesn't matter for it whatever the pH, because everything is controlled by the environment inside the delivery system.
Yes,
I
have one more question from my side.
Sure.
As an academician, what are the major differences you see in graduate students from India? They come to you. What are their skills and knowledge levels and compared
So-
to the people of your, the country, the natives?
So I think for Indian students, the students are really strong in their basic like math skills. They're extremely strong. Um, their science, chemistry skills are extremely strong. Conceptually, Indian students are really strong because they have a better understanding. When it comes to application, I see in US they are more practical. The students, for them, okay, give me a problem, I'll try to find a solution. Or for them, you need to give them in application majority of the time. Once they know why we are using it, they will understand and they will go back to it. So that's what I one another thing is, I think the communication skills is another probably a barrier to the Indian students. I would say uh, they can speak in good English, but sometimes it's not that is enough. It's you need to even I no, I have a very poor background when I started from India. I studied in Telugu medium until my seventh, and then I went to Karnataka. My parents moved to. I had to learn Kannada, and I have to do in Karnataka study in Kannada medium. So English was not my first choice at the beginning, and then in B Pharmacy, back in India at the time, 25 years back, we have a not many people they speak good in English. After I came to US, I had to struggle a lot. I can speak little English, but the communication, the way we communicate, I think that needs to be little up. I mean, I'm not saying all the students are like me. Some of students they are terrific communicator. They're really good. But I think one advice I would give to the students is not just learning vocabulary, just putting it right words at the right time. That is it, and explain and able to talk to people when you are presenting. Um, my biggest fear until even 10 years back is to just go in front of the stage and speak to the class. I would get nervous. I think we have that nervousness in Indian students. Now probably it's changed, but this is when I was studying back in India, because I moved to US 20 years back, almost 20 years back. So now I don't see much. I don't interact with not many students back in India, but those who are come to US, they already have good skill set. Any other questions?
What is actually the student faculty relationship in US, especially say, for example, how they in India, you know the difference, and you know what we the teacher expectation and the student teacher relationship and all. How is it in US?
So they respect the faculty, but at the same time, they also expect the faculty to respect them as individuals, no matter what the grades are. First, you need to treat them equally. Like you need to treat all the students at the same manner. One student, if he's really good, you cannot differentiate them. I think that's one thing. And also, they are very independent here. You have to tell them what they are supposed to do, and then they don't like once you give them the task, they want your expectations to be very clear. Whatever task you are giving, you should be very clear about your expectations. That's what they expect the most. Once you give them, they will try to do the best they can. If they cannot do it, they'll come back to you, they'll come back to you and say, Dr. Bapatla, I cannot do this, this is the reason. They'll be more upfront. And so far I have been a very good relationship with the students. I did not have any issues. I mean, I received Teacher of the Year award from different universities. So I think I have a positive interaction with the students so far.
One last question, Dr. Satish, I can proceed. How much time do you really spend on formal teaching, especially in undergraduate students as well as the postgraduate students?
So right now in this university, we only have a graduate student, like the doctorate students, Form D students, no undergrads or no masteral or no PhD students. So since I'm a department chair, I do teach about 50 to 60 hours a year, and then I have some more responsibilities on administration, and then I do also have to do research. That's part of my portfolio, and then administration and service.
Thank you, Dr. Rajesh, for your excellent presentation. It is well received. We are pouring in the responses, and we edited a few of the important questions. We will send it to you across for your opinion. I thank
Sure.
the opportunity given by the organizing committee to speak few words in this. Now I will hand over to Dr. Satish for the next proceedings.
And I would like to thank everyone.
Thank you, Professor Nagaraj Garu, and thanks a lot, Dr. Rajesh. It was an excellent presentation on behalf of Bapatla College of Pharmacy. We thank you for this wonderful session. Thanks a lot, sir.
Thank you, everyone. It's really nice to meet you. Thank you.
Have a good day, sir.
You too.
Yeah.
Thank you.